Welcome to the Temporal Graph reading group today. We are glad to host Alex Jenkins, uh, and who will be presenting their recent work, learning to predict global atrial fibrillation dynamics from sparse measurements, um, and some information on the speaker. Alex is from Imperial College London and is a PhD student there who is researching graph and time series signal processing with applications to um, cardiac fibrillation under the supervision of Professor Danilo Mandic and Dr. Fu Song Yun. And he obtained a master's degree in physics from the University of Manchester, and he is also currently a visiting researcher in the Graph ML group in Switzerland, and supported by the Hassler Foundation grant and the AI for Health Center for doctoral training in London. So I will hand the mic to you.
Okay. Well, thank you very much for the kind introduction, Andy. And yeah, it's a great pleasure to give this presentation today on behalf of all our collaborators at both at Imperial and at UZI in Lugano, Switzerland. So yeah, we're going to talk about learning to predict global atrial fibrillation dynamics from sparse measurements. So just a bit of background to start off with. What is atrial fibrillation? So atrial fibrillation is what's called a cardiac arrhythmia. In fact, it's the most common cardiac arrhythmia, affecting 1 in 3 people in their lifetimes. And this accounts for 37.5 million people globally, with this number projected to rise by 60% in 2050. It's a major cause of stroke, heart failure, and death, and a major cost to global healthcare systems. But to understand atrial fibrillation, I think for a moment, let's consider a healthy heart. So the heart is made up of 4 chambers, and we're interested in the top 2 chambers, which are called the atrium. In the atrium, in a healthy heart, we have this SA node, which we can think of as the source of electrical activity. This is sending a single organized wavefront around the atrium chamber and towards the AV node. This AV node is acting as a detector as well as a transmitter. So it sends a signal in response down to the ventricles, which then contract and pump blood around the rest of the body. However, in atrial fibrillation, this pacemaking of the heart breaks down. So in AF, the SA node is no longer the dominant source of electrical activity, but instead we have other multiple disorganized wavefronts propagating around the atrium. And these, in response, send signals to the AV node and to the rest of the ventricles to contract rapidly and irregularly. So if we have a look at the ECG waveform here in a healthy heart, we're all used to maybe seeing this sort of standard ECG waveform, which is both predictable as well as regular. But in AF, we see that the regularity breaks down, so there's a difference between timing between each of these beats, as well as the heart rate has increased here also. So that's a bit about AF, but now let's take it to an even deeper level. So let's go down to the local level of what's happening in terms of the propagation dynamics. So if we consider now what's happening at a cellular level, we can imagine part of the atrium is being made up of different cells, and these cells are each connected to each other through the geometry the atrium presents. What we have to control the propagation of voltage through this tissue is some form of the cable equation. So in this cable equation, we have a time derivative of the voltage at a given cell, V. How that changes as a function of time depends on voltage signals coming from its neighbors in space via some anisotropic diffusion through some diffusivity tensor, as well as different variables at the same cell. So these could be different measures of current, for example, as well as the conductivity of the cell, the capacitance of the cell, etc. And so just to break this equation down a little bit, what we have is these neighboring spatial regions, which are electrically coupled. We have this anisotropic diffusion term, which governs the dynamics, as well as we have this local diffusivity, as well as these local conductivity and capacitance, which can control parameters at the single cell level, but also alter the global solution, which we see for voltage over space and time. And what we see is this manifests in the cell as an action potential, which is the same as what we also see in neuronal spiking in the brain. So now if we zoom out, so we go from the local perspective now to the global perspective, as what we see at a higher level, we see for AF, like we also see in AF, a spectrum of different dynamics is existing across patients. So on one side of the spectrum, what we see is very organized beats, very organized waves. So we have this mother wave, which is maybe a rotating spiral, and this spiral rotates maybe at a fixed rate, in which the electrical excitation changes roughly in a circle. And then on the other end, we have highly disorganized types of wavefronts moving through the atrium. We have maybe multiple wavelets which are colliding and changing and are moving quite chaotically. And we find that these types of dynamics and this spectrum from organized to disorganized depends on properties of the tissue, like we saw in the cable equation. So we have the amount of coupling between adjacent regions of tissue, which is known as gap junction coupling. But we also have the pattern of fibrosis in the tissue, which is essentially also the conductivity and other parameters like this. And what we see is that they can manifest as a broad range of global dynamics in patients. So let's move on to how do we actually treat this? So now we know what atrial fibrillation is. How do we treat it? So the most common treatment for AF is known as catheter ablation. So catheter ablation is a minimally invasive procedure. The patient comes into the hospital, into the catheterization laboratory, and they're anesthetized, they're put to sleep, and a small incision is made in the groin area in which some catheters are inserted through the vein and into the interior of the heart. So here we see on the right-hand side here, we see two catheters which are placed into the atrium. So we have an ablation catheter as well as a mapping catheter. The mapping catheter is responsible for measuring the electrical activity across the surface. And what we have on this mapping catheter is a few electrodes which can sense these measurements. The ablation catheter, which is responsible for the treatment, it aims to burn or freeze parts of the atrium tissue in order to destroy the conductivity of these cells and stop potentially waves from propagating in certain directions. And kind of like how an electrician might fix an electrical problem, the clinician here can be sort of viewed in the same way as burning parts of the atrium to restore order to the heartbeat and get it beating again and paced like a healthy heart. However, we can't really map the electrical activity in the atrium correctly. And there's a trade-off which exists, which I'll go into the next slide, but this limits where we can burn in practice. So how we do this in practice is we burn the same region for all patients with AF. We burn around what is the pulmonary veins. So these are these small veins shown here on the diagram. If we burn around these veins, we see that 50% of patients, they benefit and they get cured at 5 years, but the other 50% don't, and it doesn't work. And at the moment, this is our best evidence-based clinical method for treating these patients. And this is probably due to us using a one-size-fits-all treatment approach to treat a heterogeneous disease. So we know that there exists this spectrum of possible dynamics that could exist, maybe not caused by signals coming from these veins. So what is stopping us? Why can't we treat based on the electrical activity which is actually in the atrium? And this is because we can't measure it at the moment with the coverage, so we can't see what's happening for the electrical signals everywhere at once across the atrium, but also we can't see it with a high enough resolution to precisely guide the clinician in where to burn with this ablation catheter. So just to explain what this trade-off looks like in mapping at the moment, on one hand, we have a mapping catheter, which is known as contact mapping. So this involves a grid of electrodes fed into the atrial chamber. And this grid is able to be placed in contact with different regions of the atrial surface. And when in contact, it can precisely measure the electrical activity as a time series at these points. This is the standard clinical approach used in pretty much all procedures around the world. However, this catheter, due to its finite size, it has limited coverage, so we can only see roughly 10% of the atrium per time. And in order to get electrical measurements from across the whole surface, we then have to sequentially scan this mapping catheter around the surface, collecting measurements from the different regions and trying to interpret them. One way of interpreting them is to stitch these recordings together. However, we can't do this in AF. AF is dynamic, each beat is different, and so trying to stitch these recordings at the moment together simply does not work. We can do it in some more orderly beats, some more organized beats such as tachycardias, but in terms of fibrillation, we cannot stitch these together at the moment. And so we have to burn this same spot around the pulmonary vein. Okay, so that's contact mapping. On the other side, we have non-contact mapping. So this is a different type of catheter. It's inserted into the center of the chamber and is positioned kind of in the center of the chamber. And from these points, it records a fuzzy measurement of what's happening around it on the surface. And so through some inverse reconstruction algorithms, these time series sensed from the center can be inversely mapped onto the shell of the atrium using some physics principles. What this does, non-contact mapping, is it enables global coverage of the atrial surface, so we can see a possible solution of the electrical activity across the whole surface simultaneously. So we have a nice video playing of the dynamics. However, this doesn't have the resolution required, like contact mapping catheters do, to precisely guide this ablation catheter on where to burn and where to cure. And so it's not routinely used, and it's costly. And so we revert back to our standard clinical approach of collecting these limited measurements within the clinic. So what we want to do in this work is we want to have a look at how can we transform these sparse measurements collected through contact mapping into a global map like this on the right. Being able to do this will link. Okay, so being able to do this will enable us to work with the standard catheters, which are collecting measurements every day clinically, but also it will enable us to see more and stitch these recordings together with these precise measurements made with these catheters. And so to do this, what we can imagine here is that if we have a matrix of space versus time, and we have our catheter moving around space and measuring observations for a certain amount of time, we can imagine this problem of predicting the remaining signals as a kind of matrix completion problem. So here, what we would want to do is use these signals in the observed patches with this contact mapping catheter and use them to predict the remaining 90% of the atria, which we don't see. And so in order to do this, what we can also leverage is the fact that we know some relationships in space. These relationships of each of these possible locations in space is that we know there's some coupling through the atrial geometry. So we know that there's a graph which we might be able to leverage as an inductive bias in the processing to help us complete this task. However, to solve this task, we need to also address some key challenges for AF imputation. So this contact mapping catheter only sees about 10% at once. And so it is extremely challenging to go from 10% to 90%. So how do we propagate this information in the correct way from these observations to the areas in which we wish to predict? And how do we manage some additional unknown parameters during this whole process? So at the moment, we're just measuring voltage. We don't know any other properties of the tissue. These are unmeasured. And so we would like to learn these on the fly as we perform our task. In addition, do we train a single model on a per patient basis, which would be very expensive? Or do we try and find a way to personalize a single model to perform this task across patients? And again, how do we efficiently train and adapt this during test time as new patients come into the clinic in order to achieve these personalized imputation maps? In addition, what is of key importance as well is how do we quantify the uncertainty in our reconstructions? If we're predicting 90%, we would like to tell the doctor how certain we are in these predictions in order for them to guide a clinical interpretation accurately. So our solution for imputation mapping is known as FibMap. So FibMap is a graph recurrent neural network for the task. And what we do here is we leverage the toolbox given to us by graph deep learning and graph signal processing to firstly represent the atrium of each patient as a graph. And then what we do is we represent as a time varying signal on this graph the sparse electrical measurements collected with the mapping catheter. So what we end up with here is blocks of observed time series across space as well as across time. And what we would like to do is provide these as input to a model, a spatial temporal graph neural network, to complete this matrix completion problem. We would like to predict the missing signals at times in the past and times in the future in order to gain a comprehensive global map of the atrial fibrillation dynamics from these sparse measurements, which are collected every day in the clinic. So in order to do this, we leverage a 4-step process: personalize, encode, process, decode. So I'll dive into each of these steps now. So our first step is to personalize the model. So FIDMAP is a single model, and it's trained across patients. And what we're tackling here through personalization is how do we take this single model and personalize it to give predictions at the per patient level. And to touch on this, a graph recurrent neural network is inherently a global model. So it shares parameters across space as well as within a time window. And these global models may struggle to capture elements of the dynamics which are both at a local level, so we might have node-specific dynamics, but also at a per-patient level, we might have patient-specific dynamics. And so if we didn't introduce additional parameters to capture these local dynamics, then a naive graph recurrent neural network would maybe brush over these differences. What we know from the literature is that node embeddings could allow for a hybrid global local approach in practice, and that is what we leverage in this work. So what we have is our single graph recurrent neural network model, but we personalize it by adding these embedding parameters. So we introduce a table of embeddings on a patient level, and these aim to capture properties of the patient which are unmeasured at the time of this mapping procedure. So this could be blood pressure, but also any other combination of parameters which are important for the task, which we will learn from the signals themselves. But also then we have node level parameters, and these aim to capture latent properties of the tissue, which we also don't measure, but we know are crucially important. So in our spectrum of electrophenotypes which exist, we know that the conductivity in the tissue, as well as the diffusivity, etc., needs to be captured in order to accurately describe the solution on a per patient basis. And so what this ends up looking like is node embeddings introduced for each patient. So we have an embedding table of n, which is the number of nodes in the graph, and q, which is the dimensionality of the embedding. And then we also have a patient embedding vector, which is of course of dimensionality r, and we can tune this. And this aims to capture the global dynamics. In addition, these embedding parameters may provide some interpretable insights for electrophenotyping, which we'll look into shortly. So the encode, process, decode framework is like so. So we formulate FIDMAP as a bidirectional graph recurrent neural network to perform imputation at each step. So to do this, we firstly encode our inputs. What we consider as inputs is our sparse electrical measurements across the surface of the atrium in time and yeah, of course, space, as well as we define a mask to identify if this electrical measurement is valid. So is it contained within the region defined by our contact mapping catheter, or is it not? In addition to these inputs, we provide patient embeddings and node embedding parameters in a way to amortize these local parameters into the global model here, which will apply over patients. So once we have an encoding into a hidden representation H at a time step t, and this is a vector over all the nodes, what we have here is we process it through a gated graph recurrent neural network. So what we're doing here is we're coupling together the gates of a GRU RNN here with a message passing neural network to operate and update these gates. And so essentially what we've done here is this is known as a space and time model in the graph deep learning framework. And what we've done here is we've coupled together the spatial and temporal processing in order to update this hidden state at each node and over time. And then from this hidden state, what we do is we decode into a prediction. We use 2 steps here. We firstly perform some initial predictions from H to the signal value Y, as well as we then regularize these predictions on the graph to update them and refine them. Our outputs are actually different conditional quantiles of the time series we're trying to impute. And we do this because we want to predict also the uncertainty estimates. By decoding to a set of quantile values, we can utilize a quantile regression loss function to tune and optimize our model here. So I just want to touch upon some of these parts and components of the graph recurrent neural network and draw some analogies with the local physics. Because what we're doing here is, well, what I want to explain is why the graph recurrent neural network representation we use here works well in practice. So what we have here in the cable equation is these neighboring spatial regions being coupled, and we express this through the graph topology if the patient is coupling. And then we have this term here, which represents the anisotropic diffusion, and we capture this through anisotropic message passing functions here. So the message passing functions we implement depend upon the hidden states, both at the sender and at the receiver nodes. So we can assess whether to update based on the different directions in which these messages are coming from. Next, if we look at these local parameters, so we have the diffusivity as well as the conductivities and capacitance, and we know these affect the global wave patterns. What we do is we try and capture these aspects of the model with these embeddings over patients and nodes. And these threshold-dependent activation and recovery dynamics we see in the action potentials, and we hope to capture these through the nonlinear functions in the gated recurrent neural network, but also the message passing. And although these are operating at different scales, you can kind of draw an analogy with the different components between the actual governing dynamics and the graph recurrent neural network we employ for the task. Okay, so with the model discussed, we can now discuss how we train as well as what data we use. So our data set here, it consists of 51 AF patients. And what we use to train this model is non-contact recordings. So this is the example where the mapping catheter is placed in the center of the atrium and maps a solution onto the target via inverse reconstruction. So using these recordings here, it enables us to get realistic examples of human AF dynamics to train our model, despite not being optimal in terms of the resolution they use. These recordings, they consist of fully observed, so global dynamics recorded for about 5 to 20 seconds at a resolution of 3,500 nodes and 3,000 hertz. The reason why we trained this model on these non-contact recordings, despite the limitations in resolution, is that it avoids us using some simplified mathematical models if we were to simulate the dynamics themselves, because what we observe in humans is known to differ substantially from these debated mathematical models. Okay, so now we have this data set of these global dynamics. How do we train our model? So what we do here is we develop a self-supervised learning approach in which the contact mapping is simulated from these non-contact mapping measurements. So what we do here is we simulate this multipolar catheter being placed on different regions of the tissue, and we simulate it as a random walk. So we have these this catheter moving around randomly, but it's a self-avoiding random walk, so it won't sample the same location twice. So this we can sample different paths, as well as we can control parameters here, such as the amount of time spent in each location and recording measurements for, as well as the area of the contact mapping catheter, as well as the overlap between adjacent regions in which we're sampling from. And so we can sample many different random combinations of these at training time. And in doing so, we can make our model robust to these different variations we might see in practice. And in order to optimize our model here, so we have these global non-contact recordings to train it. And during training, we want to fully utilize this information. So what we do is from this small patch, this 10% observed, during training, what we aim to do is learn a function to map from this small patch to the whole atria global signals. And so we define a whole atria reconstruction loss during training to optimize our model. And at the end of training, what we end up with is a pre-trained FibMap model. However, during test time and for new patients, and if we're going to apply this in a clinical scenario, we're only ever going to see the small patch of recordings at these roughly 10% patches. And so in order to apply this model and apply it to new patients as they come through, we want to set up a way of adapting this model. And so how we do this is through fine-tuning. Specifically, what we want to do is set a majority of the parameters and fix these from training. So these are all of the GRM parameters, the encode, process, decode parameters are all fixed at test time and as new patients come along. However, and this makes the problem a bit simpler because as new patients come along, we just want to learn the patient embeddings as well as the node embedding parameters. And this will enable us to adapt our model efficiently at test time by only updating a small subset of the total parameters. However, because we only see these small patches, we want to be able to update these parameters just using these patches. And so we develop an observed patch reconstruction loss during fine-tuning to update our model. And this is, of course, different to in training because we don't have access to the global signals for new patients in the clinic with the real measurements we're recording. So that is our dataset training approach. Next, I can show you some results. So let's start with the fine-tuning and the validation of this. So here, what we have is a test set of patients, and what we would like to do is to obtain this global solution, this imputation map, just by updating the patient and the node embeddings. And what we find is that, so this is on the validation set. And what we find on the validation set is that our reconstruction losses, as we fine tune our patient and node embeddings, our reconstruction losses converge, not only in the observed patch, which are the measurements we see, but also in our predictions across the rest of the atria. So just by optimizing our parameters based on the signals we see, we also get better predictions across the whole rest of the atria. The fact that these reconstruction losses are correlated in our validation set means that we can optimize our observed patch loss as much as possible until convergence. And it seems that our whole atria loss will also proportionally decrease as well. So we'll obtain better solutions just by using our fine-tuning setup. Okay, so that was
just a quick question.
Sure.
Is it still possible to apply the model if you don't do fine-tuning? Supposedly the clinical, like the hospital don't have the GPUs to run the model, or they just want to apply it.
Yeah, so good question. So at the moment, it wouldn't be possible. I mean, you could get some approximate predictions, but I mean, just a small amount of fine tuning will make these maps much more accurate. So if we look at these losses, so this fine tuning procedure to 100 epochs takes 22 minutes per patient. But even a small number of epochs, so say 20 epochs, we can still reach a very good value in terms of our loss here. So, and this corresponds to roughly 4 minutes. So 4 minutes of fine tuning, we can get this loss to a pretty low level, and maybe we haven't tested it, but maybe to a level of pretty good quality in terms of the predictions. But in terms of making it more simple at inference time, at test time, there are different methods we could employ here, which I'll discuss later.
Okay. Thanks.
Okay. Yeah, and so on the test set now, what we've done here is also introduce some other baseline models for this task. So this task before was, we're the first to operate on this task, so we introduced some benchmark models here. We have mean predictions, which just give the average voltage value at each node and time. So this is pretty poor in terms of reconstruction. We also have an RNN, which is trained to kind of perform imputation via forecasting to propagate this information forward. And then we have this bi-RNN here, which, like our model, would propagate information in both ways, but just at the node level. So it's not taking into account information across space as well. And then we have matrix factorization, which you're probably all familiar with, is the classic method of completing, doing these matrix completion problems. And then we have our FibMap method here. So first we evaluate in terms of mean absolute error of the reconstruction of the remaining 90%, which was unobserved. And we see that we achieve a 210% improvement upon matrix factorization here. And then also in terms of a clinically relevant assessment to use here and way of quantifying the dynamics is we look at how accurately can we predict these what are known as phase singularities in the remaining 90%. And these phase singularities, I'll show in the next slide, positions in which electrical waves rotate about. And these are characteristic of fibrillation and are thought to sustain the different types of fibrillation we see and can and have been targeted via ablation in the past. So these are clinically relevant features in the dynamics. And what we can see here is we achieve an order of magnitude improvement in the true positive rate for detecting these phase singularities in space and time. And so these are better visualized through some spatial reconstructions here and temporal reconstructions. So here each row represents a patient. So if we consider the top row first, we have this partial map, which is obtained with our contact mapping catheter. We have this true map, which we treat as a ground truth, which is our non-contact measurements. And then we see our FibMap reconstructions here. So these are in phase, these are measured in phase. And this gives a nice view as to where different parts of the tissue are in its oscillatory cycle. And what we can see is how these wavefronts move around using phase as an example. So here, what we can see is our FibMap has reconstructed these multiple wavefronts traveling in different directions, which were otherwise not visible in the small patch observed with the multipolar catheter. We also add in matrix factorization here as a baseline comparison, and we can see that clearly the dynamics have not been captured through this very simple approach. On the other hand, for this other patient down here, we have some rotational activity, which is being observed in the ground truth map, but also predicted by FibMap. And this is what we're calling the phase singularity. The phase singularity would be this position right at the center where the phase is undefined. It's kind of where all the different phases meet. And so it's the center of which these spiral waves move around. And we can see that clearly we've been able to reconstruct this point very well in space and time, despite not seeing it in the original patch of measurements. So that's what the solution looks like for a snapshot. This is, of course, a video. So to give an idea, an indication of what these predictions look like over time, we consider the time series at a single node. So we have FibMap this at the top for these two patients, as well as MF down the bottom, which is matrix factorization. And clearly here you can see that FibMap has reconstructed the ground truth, which is shown in black, very well in terms of amplitude and both phase. In regions in which the amplitude varies slightly, you can see that using the quantile regression and our predictions of the uncertainty intervals have been able to capture the amplitude variations quite well. In comparison, we see matrix factorization is broken down in terms of both phase and amplitude, and clearly has not captured the inherent nonlinear dynamics present in these signals.
Alex?
Yeah.
Yeah, these time series, they are what each node gives, and what each node has shouldn't be like an ECG. Oh no, it's not an ECG. Like, what is it exactly? This is just like what this area membrane voltage is.
Yeah, so let me explain what you're seeing here. So this is a single location of the tissue, which you can imagine we've placed this catheter on, and we're measuring it with an electrode. So we're in contact with the tissue, and what we're measuring is the voltage. So this is the voltage changing as a function of time at this particular location which we placed our electrode here. And we can see this blue patch. What this is meant to represent in the middle is the duration of time in which the electrode is actually placed there. And then it moves to a different region somewhere else, as we're mapping around with our catheter. And outside of this blue box is our predictions, given this information as well as the other patches in space and time to perform this reconstruction. But yeah, what you're seeing here is it's not an ECG. It's known as an intracardiac electrogram. An ECG is, of course, from the outside of the body, but this is in contact with the.
Yes, yes. And if you take like the maximum like intervals between the maximum peaks, this should correspond to the RR intervals, or like, because like for example there are peaks that are like towards the end of the observed times, there are like smaller peaks that seem like
Mhm.
like for example 2 peaks before the observed times, right? You have yeah, can you go for the mouse
Mhm.
to,
Maybe these
yeah,
ones here.
these ones for example.
Yeah. So yeah, this is it's kind of not the same as an RR interval of what you see on the surface ECG, but it is an indication of so what is kind of happening in the cells which are captured with this electrode. Captured with this electrode, there's many different cells and they're all firing, and so we kind of see this voltage as kind of the is kind of like the accumulation of all of these different signals, and they've all been summated here. And you see that there's some of them, maybe half of them, a peak, a kind of deeper repolarizing here, but the other half of these different cells are doing something else, and so it might smooth out. So really, this voltage is many different cells, which we're seeing. It's still very precise and localized, but the cellular level is, of course, much smaller. So some of these peaks might be very neat, and it means that all the cells in this area are very synchronized, but here they're quite desynchronized, I guess. So that's why you see it's quite flat. So yeah. Okay, so yeah, with these results discussed, I'll move on to some of the other insights we can take from our model. So what we've looked at here is what do our hidden states in the in FibMap represent, and do they capture any properties of the patient? And when we analyze these state space dynamics, so this is the vector which is updated by the GRNN at each node in time, and we plot these for a single node in an embedding space. So we reduce the dimensionality to 3D here using t-SNE, and we can see that these states resolve sort of rotate around fixed orbits within this state space, and this is sort of like an attractive dynamics. This is what we see, and is characteristic of chaotic dynamics. And so our first impression upon visualizing these was that our GRNN can capture the chaotic dynamics of AF in terms of these state space dynamics. Also, that what we can do here, as seen in this recurrence plot, is we can also analyze these different state spaces at the different nodes across the atrium. And what we can do is we can see if a region of state space is revisited sometime later. And this might indicate as to how predictable the overall signals are. And we can, of course, do this on a per patient basis and reconstruct these plots of time versus time. And if they recur in a similar region of space, we get a black dot here, white otherwise. And you can see that, yeah, you end up with these different patterns which can represent basically how predictable, how disorganized the hidden state dynamics are. And so basically, some of the insights here for the state spaces could give us extra information for electrophenotyping for patients, which we haven't dived into and validated using different parameters or seeing how the patient might respond to different treatments. But it gives an indication that our model is capturing something intrinsically important about AF, and that is that the dynamics are chaotic and they do oscillate around these fixed points in state space. On the other hand, we have an analysis of our embedding spaces, so our node embeddings here. And what we've done here is we plotted them and reduced the dimensionality to 2D. So we've done this via t-SNE here, and we see these points which represent each node plotted in 2D space. The first one here is they're colored by patients. So same colors represent the same patients. But more interestingly, when we plot them using characteristics of the time series, for instance, the dominant frequency of the time series, once we've taken a spectral decomposition of it, what we see is that, yeah, it roughly clusters depending on the frequency in which these cells oscillate at each node, so with the underlying frequency at each node. So as you move up this plot, we sort of get higher frequencies within the voltage dynamics at each node. And then also we've had a look at the Shannon entropy just to see what else these node embeddings capture. And the Shannon entropy kind of captures the measure of organization, of predictability, or information content of these node level time series. And so we can capture some quite interesting characteristics of the node time series here. But also we would like to, in future work, extend this to analyzing different properties of the patient and tissue and seeing how these might be useful in a clinical context and to guide an interpretation of the type of dynamics we're seeing. What we've also done is performed a sensitivity analysis here. So because we have control over simulating these contact mapping catheters, so we've simulated as a random walk, of course, we can control the area and how long we spend in each position. And so we can get an idea as to how our reconstructions depend upon these parameters. Some simple insights here. What we've seen is that our prediction accuracy increases as the time in which we spend in each location decreases. So this means basically we're able to fit more observations across space within our time window. And so our predictions might increase as a result. Predictive accuracy might increase. But also what we've seen here is, of course, when you see more of the area per time, your reconstructions get better as well, which makes sense. Also, we'd like to just show you B on the right, so this right-hand side plot. What we've done here is plot how the reconstruction error changes on average as a function of the distance away from the valid observations. So, of course, the distance here can be measured in space and time. So we define this imputation horizon, which is essentially the number of hops on a spatial temporal graph away from the nearest observation, valid observation. And we can see that our prediction error increases and then plateaus. Correspondingly, when we plot our predicted confidence interval on our predictions, we find that these intervals also increase, which is good because we're able to now capture the corresponding uncertainty as our error, prediction error also increases. So that indeed our confidence intervals are insightful for guiding a clinical interpretation of our predicted values. And finally, what I'd like to show here is a clinical proof of concept for our method. What we've done here, and for a few patients in our test set, is we have non-contact measurements performed. So we have the global dynamics across the atria, but we also have simultaneous, on the same morning of treatment, we have these contact mapping measurements taken. And so we have this global ground truth we can use, and at some time later, we have our contact mapping recordings taken. What we do is from the contact mapping recordings, we produce an imputation map, and now we would like to compare this against our non-contact mapping data as a ground truth. To do this, because atrial fibrillation is dynamic, we have to come up with a different way of comparing these two dynamical windows of measurements. And how we do this is using a sliding window approach, in which we correlate the imputation map with the ground truth given with this non-contact mapping. And we slide this window across and we calculate how correlated these different times and patterns we see over space are. So what we end up with for each patient is a imputation map, window index against another window index representing where we are in the non-contact map. And then we have these correlated values here, which we can visualize. What we do is we flatten this into a distribution, and then we can plot this across our 3 patients, what we see. And so what we want to show here is that imputation mapping can capture personalized patterns, as well as it can capture personalized patterns, but also it can capture aspects of the dynamics we also see in our ground truth data taken some time apart. What we've done here is the blue distribution here represents the imputation map compared with the ground truth of the same patient. We can see that these distributions of correlations have wide tails, which means that there's more windows on average with a higher correlation. in these intra-patient comparisons. And then we've also compared the imputation map of the same patient with the ground truth of different patients to get this inter correlation distribution here. And we see that this is peaks at lower correlation values here, indicating that less windows have high correlations. And so that there's less windows with personalized aspects of the dynamics being captured. And then we also compare against a shuffled baseline to show that our predictions are better than random, and indeed they are. So to give you some indications here as to what we can uncover using these real-world contact mapping measurements as input, we can clearly see that we've been able to predict this wavefront propagating down in terms of this patient. And when we found the next window which correlates with this very well in the ground truth, we can see also similar aspects of the dynamics also being captured with this other modality here. We repeat this across 3 patients, and you can see that FibMap has captured dynamics here just from these 10% recordings with the contact mapping measurements. You can see what we've also captured these rotational dynamics which were present in the ground truth. And so to come to a conclusion with this talk, I would like to discuss firstly how we envision imputation mapping being used in the clinic. And so at the moment, we have new AF patient arrives to the clinic. We perform some ablation of just the pulmonary veins. And then that is the end of the current workflow. So we isolate these regions electrically, and it works in 50% of the patients. So now what we attempt to do is we could prolong this workflow. So what we could initially do is use this mapping catheter effectively now, be able to move this around the atrium. Firstly, we would use some initial measurements from this contact mapping catheter to fine-tune our patient and global patient and node embedding parameters. And then after these reach a certain level of error here defined by these curves, then we could perform imputation mapping. So now we're moving around with the catheter, and in response, a clinical team is able to obtain real-time predictions of the global dynamics here. And then, which we haven't done in this work, but would be a potential downstream use of our method now, is to then analyze these global maps and then decide on the course of treatment for the patient. So we can move beyond a one-size-fits-all treatment strategy here, and potentially towards personalized treatment by hopefully answering questions such as where should we ablate other than pulmonary veins, or maybe don't ablate at all. Maybe a different form of treatment might be better for these patients. And so hopefully imputation mapping upon further validation could enable us to move beyond one-size-fits-all treatments. Of course, there's some limitations here. So the key one is that our model is trained on non-contact data, and I've pointed out that this data modality has a lower resolution. However, in the absence of a true ground truth of fibrillation dynamics for patients, this enables us to train and validate our proof of concept of fibrillation, of imputation mapping. However, in order to move this towards being an effective clinical tool, we will need to determine the spatial temporal resolution required for guiding these ablation catheters in practice in order to accurately treat the disease. In terms of technical improvements, in order to reach these high resolutions with what we've done, potentially one could consider augmenting the training with the non-contact mapping, but also using some simulated data at a higher resolution to essentially improve the resolution of the imputation maps. Also, one could look at exploring hierarchical architectures. So this could be a graph recurrent neural network with some virtual nodes positioned in this in order to improve the long-range predictions across these very sparse set of observations which we collect. And then finally, in terms of the clinical validation, we would like to validate our model's state space and embedding representations and see what information is truly held within these for electrophenotyping patients. Of course, our understanding of the physics tells us that these local parameters capture some and, of course, determine what we see globally as our solutions. So there might be some information contained within these which we would like to potentially exploit. In addition, we would like to validate, of course, what resolution our maps would work at best in practice. And to do this, we could form imputation maps for diseases and arrhythmias in which are already stitched together quite accurately, and we could assess the differences and present them to clinicians to find out how best to circumvent these limitations of our approach. Finally, what we would like to do in the next few years is, and probably the ground truth to tell us all of our, if this could actually be applicable in the clinic, is perform some sort of clinical trial. And we could assess against a baseline of the one-size-fits-all treatment whether the information provided in the invitation map could enable us to perform better than this baseline. And so with this, just to summarize our key contributions, we've introduced a novel imputation mapping approach for AF. We've seen a 210% improvement over baseline methods in terms of mean absolute error reconstruction. We've seen an 11 times better phase singularity detection, which is predicting key properties and interesting properties of the fibrillation dynamics. We've been able to adapt fast to new patients at test time, allowing for a feasible solution within the duration of the mapping procedure in the clinic. A method for uncertainty estimation to guide the interpretation, as well as a real-world proof of concept with our data. In terms of the clinical impact, this would integrate seamlessly with existing clinical catheters, which are used daily around the world. Also, it would reveal AF mechanisms not visible within the patches of observations, the sparse patches. And in doing so, we have the potential to reduce the procedural time, allowing the doctor to visualize the activity quickly, as well as reducing the procedural complexity and the risk of burning areas in which they shouldn't. All in all, imputation mapping offers a pathway towards personalized AF treatment beyond one-size-fits-all approaches, which could raise the success rates of current treatment strategies. So with that, I would like to thank our collaborators on this project and thank you all for listening, and open for any other questions you might have. So thank you.
Yeah, so thanks a lot for presenting. There's one question from from Julia. Do you want to ask it yourself, Julia, or do you want me to relay? Oh, so there's some connection issue on Julia's end, so I will just read the question. So where did you get the data from? And is this publicly available data set, or is it a clinical trial data set that's just created for your paper?
Yeah, so unfortunately, this isn't an open data set. So it's from actual clinical measurements taken within the hospital I work at Imperial College. And so we haven't got the ethics, unfortunately, at the moment to share this more broadly. Although this is something I'll have this conversation with my PI, and we can try and act on this. If this is something you're interested in looking into yourself, we can try and get something underway.
Yeah.